Our next speaker is Stephan. Yeah, I pronounce it on German. Stephan Sacher. Stephan is a principal scientist at Research Center Pharmaceutical Engineering GmbH. Most of you may know it as RCPE in Graz, and he's leading the continuous manufacturing and PET group at RCPE. He studied process engineering at the Technical University in Graz and obtained his PhD in the field of particle technology. In his current position, Stephan is focused on development of continuous manufacturing routes. It's about the lunch break, so I need to get back into the flow. He is author and co-author of more than 60 scientific papers and international patents, and has given numerous talks at international conferences, and he's going to add the next one to this list now. Thanks for being here. Stage is yours.
Thank you for the invitation to talk here. Let me just check how that works out. It does perfectly. Yeah. Wonderful. So my talk will focus on digital real-time release testing. In modern manufacturing lines, there's a lot of data available. We have PET equipment, we have also univariate sensors, we have pressure, we have temperature, we know feed rates, so information from equipment. Why not use all that for real-time release testing and speed up the release procedure using that? So let's have a look on this slide. Modern manufacturing lines use continuous manufacturing, end-to-end manufacturing. We've heard a little bit about that. Modular manufacturing, especially with continuous manufacturing and end-to-end, we can talk about high-speed manufacturing. That means the throughput times go down from days or weeks to hours or a shift. But there is still a bottleneck before the product reaches the patient. So all this speed in the manufacturing process does not help if we stick on the release procedures we have established, because this can last up to in the range of months, so 45 days is such an average value we figured out. And a lot of CQAs have, of course, been tested before the product is released to the patient. One of those is the dissolution. Samples are drawn, we have to wait for the QC lab before the process can go to the next step. So one alternative would be, or is, real-time release. With real-time release testing procedures, this bottleneck can be a little bit reduced to a range of about 1 day. Real-time release testing uses data that is collected during the process from PET or from other sensors, from equipment, as I mentioned in the beginning. We need some process understanding that connects the data that is collected, so critical process parameters, material attributes, with the critical quality attributes that are when predicted, and at the end these predicted quality attributes are used to release the product. Especially when looking for continuous manufacturing, real-time data is needed. Because when we draw a sample in a process in batch, okay, we have to wait anyhow, but for a continuous process, when we take this sample, go to the lab, wait hours, the process is gone. So we have no information and we produce something where we don't know what is going on. So the next thing where we can use this real-time data is for quality check, for diversion of out-of-spec material, but in the best case we use models, we predict something that goes on, so predictive process control is the word, and avoid that some out-of-spec material is created, so when we leave a reference, a target, a set point, due to any reason, we can react because we predict something based on this real-time data. I'm not going to talk about that, but just to give you a little bit of overview what real-time data is for. And last but not least, and this is the goal of the talk today, is the endpoint testing. As seen already in the first slide, the endpoint testing can be substituted by, and has to be substituted by, real-time data, real-time CQA prediction, which is also very important in continuous manufacturing for tracking, because we have to match the material that is released at the end with all the raw material batches. When we change one raw material A to B, C, and so on, we have to manifest that for the authorities. And the next thing is the digital aspect. We've heard about artificial intelligence already. There's a lot of digitalization ongoing, but what is the objective in pharma for digitalization. There are a few different points or stages. We start with process monitoring. The first step is real-time data, as already mentioned, used for process control, for real-time assessment of the quality of the product. So here we need PAT and, sorry, not yet familiar with this thing. Wrong direction. That's the pointer. PAT and soft sensors. The next stage is when we create models. These connect the process parameters or the measurements with the quality of the product and can also be used for for tracking, residence time distribution models, for example. The next step is when the control strategy. For control strategy, it is essential to have data in a digital form that this the digital twin, which consists of the process models from the previous step, can be connected with actuators in the process with the equipment, but also gets real-time information from the PAT equipment and from other equipment. So these interfaces are essential, and when we can do process control and quality control in real time. And last but not least, digital real-time release testing. What is meant by that? That's a kind of digital birth certificate. So we collect all the data during the process, match it with the product, and at the end of the line, by pressing one button, we get the list out, this certificate, what process parameters have been seen by which part, which lot or which tiny amount of the material, and this is our digital birth certificate. So how can we realize that? Our case study has been carried out on a Consigma 25. Most of you are, I guess, aware with this equipment, but a little bit of an overview, similar to what you have already seen in the morning from Bole. This is from GEA, but similar unit operations. We start with feeding different materials in a twin screw wet granulation line, a pneumatic transport to a 6-segmented fluid bed dryer. This is very important to mention that at this line, while we are continuously running here at the granulation step, this is semi-continuously. So the material is separated into mini batches, 6 batches in this dryer, and these mini batches are then conveyed to the next process step, which is a granule conditioning unit. Sorry, granule conditioning unit here. Whereas, for example, a mill that creates the right particle size distribution, and after this granule condition unit, or in the granule condition unit, we have also additional feeders and a conical blender, where an external phase, a lubricant, or whatever you want to add as external phase, is combined with the PKs. We're still talking about this PKs, semi-continuous working, before we have here a diversion system and add to the tablet press, where we have a continuous operation mode again, and a little bit of intermixing between these mini batches or product keys. The Consigma standard configuration has not much PAT implemented. There is a lighthouse probe, NIR based, which can be implemented either in the fluid bed dryer to measure the residual moisture, or in the granule conditioning unit. The second thing is a weighing scale, where the external phase is weighed, so you know how much additional material you add to the product keys and can calculate the content of the different ingredients later on. So we expanded a little bit this PAT concept and implemented a lot of other interesting things. Here you see again the flow chart. We start with powder, have wet granules, dry granules, milled product, milled granules, and the tablets at the end. And what we implemented is first for wet granules to measure the content. Unfortunately, we had to use Raman because it's wet medium here. The next thing is particle size distribution, or particle size distribution characteristics that was measured via spatial filter velocimetry. I have to explicitly mention these are only examples for PAT tools, and I'm not talking about these PAT tools today, but about this digital real-time release testing concept, and this can be expanded to any other technology and to other CQAs that want to be measured with appropriate PAT sensors. So the next step after assessing the wet granules in content and particle size distribution, here is an example of the particle size distribution, B-model distribution, is the dry granules. In the dry granules, very important, the residual moisture. And here we have created a soft sensor. I press the button a little bit later, explaining first why this is a good point for a soft sensor. A soft sensor is a model that predicts, based on different collected parameters and data, something, a CQA for example. In our case, the CQA of the intermediates, the residual moisture or loss on drying. And the fluid bed dryer is a good example because in the fluid bed dryer you have a lot of information available. The incoming drying air is usually measured, you know the amount, you know the temperature, you know the moisture in this air. We should also know how much material is in the drying chambers and with which moisture it enters. So for a kinetic drying model, it's very easy to predict based on this available input data, the output, which is the residual moisture. And I hope I can start now this video. I try to press, maybe this helps. No, this does not help. Is it running now? Yeah, it is. So what you see on the left-hand side is the residual moisture at the different drying chambers, so for different pKs. It scrolls down now, so 6 pKs, and the nice thing with such a soft sensor implemented at this stage is that when we, for example, would use the lighthouse probe, we would only measure at one single chamber, so only 1 pK. But with this sensor, with this soft sensor, we can predict the moisture at each single drying chamber for all the 6 pKs. On the right-hand side, you see the temperature, and just a little bit of explanation what happens. So here we have an empty container or an empty chamber. The material is filled in, moisture is high, is of course reduced by the air, which is here also dropping down. You know, when we have drying, the air drops down. That is all calculated by our soft sensor. And the next pK is when filled, when the chamber is emptied, and the next cycle starts again. So with that, I proceed to the next slide, which is also a soft sensor, a model to predict the dissolution profile of the tablets. How are we addressing that? We have a parametric model. This can be a Weibull model, a Hill equation. We use the transfer function that predicts the dissolution profile via some parameters that are the model parameters that have to be parameterized by some by DOE, for example, or by experimental data. And in our case, these parameters here, T1 and T2, which are 2 time constants of the transfer function that we used, are correlated by input parameters. And these input parameters were identified as those which influence most the dissolution profile. So the critical process parameters is the first moment of the particle size distribution, which is gives information about the particle size itself, the loss on drying, which comes from the soft sensor, and the main compaction force in the tablet press. So this is the last example of our sensors. And in contrast to the conventional PAT concept of this consigma, here a summary with PAT. Here in this first stage, the API content plus the tablet weight, which comes from the different scales, the CQA, assay and content uniformity are predicted. With PAT, here the particle size distribution, and LOD and information from the equipment again, the main compaction force, the dissolution profile is predicted. And both is available digitally at the end of the line. To get that available in digital form, we have to link the acquired data with the material that passes and propagates through the line, and we have to implement all in a data management platform, of course. And the link is the essential thing that is done by 2 different approaches. We have heard that we have a continuous and a semi-continuous part or parts within the line. For the continuous, it's a residence time distribution model. This model is used in the twin screw granulator and in the tablet press. And in the semi-continuous, we have just a logic algebraic algorithm for the different pKs that tracks the propagation through the line in these semi-continuous modes. What is also important to mention is that in the tablet press, so whereas the transfer from the pK to the continuous mode again, we assume that there is no pK intermixing before we enter the tablet press. That just the intermixing is happening in the feed frame. In the hopper, we assume that we have nice mass flow and the measured concentration of the API that is measured. Sorry, again. It's not becoming my friend here. The concentration that is measured here is assumed that it propagates through the line. Of course, here nothing will happen to the concentration when we don't, except from, of course, from adding an additional mass here in the granular condition unit. But here, to the mass flow consideration, we assume that the concentration is just changing in the feed frame. The second assumption is that the pKs are aligned to the tablet properties with the RTT model in this tablet press. So, how does that look like? At the feed frame inlet, you see on the top a figure where you see the pK number over process time. So our interval is 180 seconds. In the first 180 seconds, the feed frame sees just entering the blue number 5 pK. After this 180 seconds to 360, the orange one, and to 540 seconds, the yellow one, and so on and so on. And in the tablet press, where happens the mixing, where we need the residence time distribution model to compute or predict the concentration, the content in the tablets at the end, according to what happens. Of course, this has to be calibrated. We did it via impulse response experiments with color tracer and also with concentration measurements with a known API concentration. So, and at the end, here as an example, this is the tablet of interest, and we can calculate that a huge amount of green pK is in this tablet, a little bit of the purple, less of the yellow, and only almost nothing from the first one, from the pK number 5, the blue one. And in that way, we can now predict the content in the tablets, the API content. Looking at this diagram, we have here the API content over the production time and different graphs. The blue one is the content at the entrance of the feed frame, and this is exactly what was measured with the Raman after the wet granulation, plus the computation where these algorithms, how it propagates through the line, if, for example, additional mass is added in the granular conditioning unit. We have an intentional change here that was caused by varying the feed rate. In our case, we increase the feed rate of the excipients, which of course leads to a drop of API content over the mass balance. The red line, is the prediction of our model, and with some time delay, we see the tablets coming out with this peak. The purple ones is offline measurement, samples drawn, brought to HPLC measurement, and we see this is in quite good, I would say, satisfying agreement that shows that this approach can really track the process and some events to the very end of the process to the final product. The second example of results is regarding dissolution. Again, let's have a look at the model. The parametric model predicts the dissolution profile. It gets some constants from the correlation, and the correlation model gets the input from the process. In our case, that was the moment, which refers to the particle size, the LOD, which is the residual moisture from the soft sensor, and the main compaction force. This comes from the equipment. So you see different sources of data from the equipment, from a typical PAT tool, and from a soft sensor from a model. This all can be easily combined. In the second row of the slide, we have some diagrams where we see propagation of variables over process time, or the same process time. First one is the granule size. The granule size, always in blue measurement and in red the prediction. The granule size comes from the measurement of the spatial filter velocimetry, and in red the prediction. And we see here also some event, some intentional event, and the prediction with some time delay can depict that. The same for the loss on drying, where the soft sensor, main compaction force, here we have some circles, and these circles are the real measurement of the compaction force, while when the tablet press is on, so our algorithm checks if the tablet press is switched on or not, and in case it is on, we collect these measurements. The blue one here is not a measurement, this is the reference, the target value, that a set point. But here again we have prediction and measurement. This is not prediction measurement, these are the both predicted time constants exactly from this correlation model, so the output from these parameters, the two time constants. And the nice thing is that always when something happens, for example main compaction force changes, we see a change in the time constant where we can already assume that the dissolution behavior will change. When we see loss on drying change, also a change in the time constant, and last but not least the granule size, here this drop causes also a drop in the time constants of the dissolution model. And when we now use the dissolution model with these predicted time constants for these 3 points here indicated as 1, 2, 3 and 4, we have done the prediction of the dissolution in real time, of course, and also drawn some samples and measured offline via conventional dissolution analytics. And we see, for me not so sufficient, and for my colleagues not so sufficient match, because that means we have to do some additional work, but not on the digital real-time release testing concept, rather on the dissolution model itself. Maybe this is not so nicely fitting our model substance, which was not an API, but a surrogate that was methylparaben, which we have used. However, in general, we see the trends, and for all these points here where we have different events, drop of particle size, change of the residual moisture, we can predict the dissolution in a little bit satisfying amount. So at the end, we can say that such an approach would support to speed up the release procedure significantly. And why not use data that is already available? The equipment gives us data that is available. We have PAT installed, spectroscopy and other devices, so we can collect that, condense it, align it with the material for continuous manufacturing process. This is necessary anyhow, and use that in a digital form to press the button at the end of the line and have our digital birth certificate. So thank you very much for your attention, and I'm open to questions.
Thank you, Stephan. Interesting talk, nice talk, on-time talk. Other questions to Stephan? Bent.
So you have a very strong correlation between the compaction force and the dissolution profile. And now you can predict the dissolution profile based on also the compaction force. Could you turn it around, saying all the other parameters will indicate that we will now have a change in the dissolution profile, so we'll control the compaction force based on your prediction model, and so that you will have constant dissolution profile?
Yeah, absolutely. Thanks for that question, very interesting. So first, what I have to mention, of course, it's very individually for product and process, as we all know. So in that case, the main compaction force has a big impact. And as I said, it must be an assessment of all the input parameters which are mostly affecting the CQAs. In our case, yes, the dissolution profile is influenced by the main compaction force, and we can turn it around by using models. I would not use exactly, also we would have to do, or we have such models that can do that, predict the dissolution profile based on directly the input parameters, and that can be used for model predictive control. But this would be one of the steps that I had in the beginning of the slides. But of course, this is possible. We could turn that around and influence intent marginally the dissolution profile by varying the CPPs based on the model.
Any further question? No? Oh, Manuel, sorry, yeah.
Hi, thanks for the nice talk. I just have a question, and it's about regulatory expectations. When you present that to health authorities into a dossier, the validation status of the process model, I guess this is a topic that, at least from our side, we've been discussing a lot. We don't have an answer, and maybe I was wondering you can give us some insights on that.
Thanks for that very important question. I was rather expecting that industry has more answers on that question when we do. Of course, we're also in close interaction with FDA, with EMA, in different working groups and so on. But I think such guidelines are still missing. There's a little bit, where is this EMA guideline on real-time release testing, there is some guideline, I think, or at least a white paper that I have recently reviewed on modeling over the life cycle, but there is never this correct, this discrete request how to do that. So I really cannot answer this question, and I just say we face the same issues always when we do together with partners such an implementation, and it has really been done from case to case and always based on sound science. So this is always what is, I think, the main concern, right? You have to prove it.
Okay, thanks again, Stephan.
Thank you
for your nice talk.